Hi everyone, my name is Takudzwa Madzivanzira. I'm a final year PhD student based at the South African Institute for Aquatic Biodiversity SAIAB and registered with the Department of Ethology and Fisheries Science at Rhodes University, both which are in a small town called Makanda in the Eastern Cape province of South Africa. I have 5 years experience in freshwater ecology, which ranges from fish foraging ecology, water quality, macroinvertebrates, and invasive species that I'm currently working on. And in these fields, I've authored and co-authored a total of 6 published peer-reviewed scientific articles. You can follow me on Twitter. My handle is at Taku1945, and you can as well follow our research by liking our Facebook page, that is Olaf's Research at SAIAB. The title of my presentation is Invasive Crayfish Have a Predatory Advantage over a Native African Freshwater Crab at Higher but Not Lower Temperatures. I'm supervised by Professor Olaf Weil and Dr. Josie South, and this research is part of my PhD study which focuses on invasive crayfish species that have invaded our freshwater systems in Africa, mainly focusing on 2 crayfish species that are spreading at a fast rate in the continent. That is the Australian red claw crayfish, which is native to northern Australia, as well as southeastern Papua New Guinea, and the Louisiana red swamp crayfish, which is native to southern United States of America, as well as northern Mexico. Our continent has been a recipient of 9 crayfish species, with 5 of those, as shown in the map legend, establishing naturalized populations in the world. The main driver of these introductions was to provide socio-economic benefits through aquaculture and fisheries development, but however, there is limited evidence of success. This map, which is from our recent publication, shows introduction routes of the established crayfish species from outside Africa, as well as translocations within the African continent. But why should we in Africa be concerned about these invaders? It's because invasive crayfish species have generally been shown to negatively impact native biota across different levels of ecological organizations. Also of concern is the scarcity of information on crayfish impacts in Africa, as illustrated in this graph, which is also from our recent publication. This does not do us a favor as conservationists, since there isn't enough evidence to compel policymakers against further introductions of crayfish species. Our study therefore presents a vital step towards understanding the impacts of crayfish in African freshwater systems. And to achieve this, we conducted comparative functional response experiments, a metric that quantifies the rate of resource use with resource density. Non-native species have been shown to exhibit higher per capita effects when compared to the native analogous species, and thus resources such as native prey communities are vulnerable to potential extirpations or extinctions. 3 categories of functional response types have been classified by Rolling 1959 as illustrated. Type 1 functional response describes a constant proportion of the available prey consumed, irrespective of prey density, or a linear increase in feeding rate. This type is predominantly found in filter feeders as they are not limited by handling time. Type 2 functional responses is characterized by a decrease in predation with an increase in prey availability. This relationship suggests high resource utilization at low densities and is associated with an inherent risk of resource depletion because rare resources are used at high rates. For the type 3 functional responses, the predation rate decreases at both the low and high prey densities, providing a sigmoid relationship. To model the functional response type, the R package FRE is used using maximum likelihood estimation and Rogers 1972's random predator equation. The main consideration of functional responses analysis is acquisition of the attack rate, handling time, and maximum feeding rate parameters. And in cases where it's problematic to compare 2 functional response curves, A functional responses ratio, that is FRR, by Cuthbert et al. 2019 is used, which combines functional response parameters in order to elucidate potential intermediate impacts. For the comparative functional response experiments, we used 3 decapods, that is 2 invasive crayfish species and 1 native crab of the Potamonatus genus. This is because the freshwater crab is the only species which is close to crayfish, as our freshwater systems on mainland Africa are devoid of native crayfish species. We collected the red claw crayfish from the invaded Inkomati Basin in Mpumalanga province, the red swamp crayfish from the invaded Mimosa Dam in Free State province, and crabs from dams around the Eastern Cape province. We used sharp-toothed catfish, that is Clarius gariepinus fry, as prey, which we purchased from Aquaculture Innovations here in Makanda. Clarius gariepinus is ubiquitous across the invaded ranges in Africa, and it is a commercially important aquaculture and fishery species, and the behavior and morphology of their fry is similar to a tadpole and thus also representing an amphibian proxy. We performed functional response experiments in rectangular tanks with those dimensions in a controlled room at SAIAB. Measured decapods for the experiments were starved for 24 hours, that is to standardize hunger levels, and we allowed them to acclimate to experimental arenas for an hour before prey was supplied at those densities, that is 2, 5, 10 up to 200. Experiments were run for 3 hours, after which the predators were removed and the number of prey items consumed were enumerated. 2 functional response experiments were completed. 1, under light and dark conditions, which represents daylight and night times respectively, and 2, under 19 and 28 degrees Celsius in dark conditions. These temperature treatments fall within the temperature ranges of freshwater systems in Southern Africa, as well as the ranges that these decapods tolerate. Prey survival was more than 99% under all control treatments, and there was no mortality of predators during the experimental period. All the treatments and combinations resulted in a significant type 2 functional responses, which was higher under dark conditions than under light conditions, as illustrated in these 2 figures. This was driven by consistently higher attack parameter values and lower handling parameter values in the dark treatments. Also from the dark treatment, as you can see from the initial slope steepness of the functional response curves, both crayfish species had higher attack parameters than the crab. However, the low handling parameter values for the crabs and the red claw crayfish under dark conditions resulted in maximum feeding estimates as well as high FRR values. When comparing the 2 temperature treatments under dark conditions, crabs had the highest maximum feeding estimate, driven by low handling time at 19 degrees Celsius, but however, the FR curve is not significantly different from that of the red claw under the same temperature treatment and at the highest prey densities. Peak Lucky had a long handling parameter at 19 degrees Celsius, which resulted in a low maximum feeding estimate as well as a low FRR value. In contrast to the 2 invasive crayfish species, when temperature was raised to 28 degrees Celsius, the crab FR magnitude significantly decreased, resulting in the lowest maximum feeding estimate for this treatment. Both crayfish species had higher attack parameter values at 28 degrees Celsius, which resulted in high maximum feeding estimates as well as high FRR values. The consistently higher functional responses in the dark compared to the light maintains that all 3 species are feeding more during the night. These decapods detect prey through chemosensory pathways and therefore do not really rely on visual foraging cues. Also during the night is when crayfish cause havoc to the artisanal fishery, which has been reported in Southern Africa. Fishermen in Southern Africa, for example in the Zambezi Basin, set their gill nets just before sunset, and retrieve them just around sunrise. And during the night, crayfish spoils fish caught on the gill nets by partial consumption, resulting in unmarketable fish as shown in this picture. The crayfish also damage and entangle themselves in gill nets, which results in reduction in efficiency of that net, and in most cases, the nets are discarded. Since freshwater crabs are also nocturnal, they are likely to interact and share common resources with crayfish during the night when both species emerge to forage. This could result in competitive exclusion or elimination by predation of the inferior species. And we already have reports on freshwater crab declines attributed to peak laki, for example, in Lake Naivasha, Kenya, although the impact mechanisms are not clear. Temperature treatments had dissimilitude effects on the handling time of the three species, whereby the per capita predation by the crab and red-claw crayfish consistently exceeded that of the red-swamp crayfish under almost all the experimental conditions, apart from when we raised the temperature to 28 degrees Celsius. Our results support the ecological matching hypothesis, which intones that the performance of invasive species are higher at temperatures that closely match their thermal optima. And in any case, both invasive crayfish species have a competitive advantage at higher temperatures compared to the native crabs. And further, due to the predicted climate warming, the invasive crayfish may potentially establish in previously unsuitable habitats, thereby increasing their invasion ranges. The high FRR values for the two crayfish species are of concern, as they show the potential impacts of the two crayfish species on benthos, here represented by Clarius caripinus. These impacts on benthic fish have also been documented in England, where the presence of signal and rusty crayfish resulted in benthic fish mortalities due to direct predation. Appreciable predatory impacts of crabs on native fishes have not been recorded, although in this study, the functional responses for crabs was higher at 19 degrees Celsius. The invasion by crayfish will, however, result in additive pressure on resources that were previously not threatened before the invasion. And in future, if the decapods coexist, the pressure on native prey would continue, which could lead to extinctions of native species and ecosystem destabilization. The differences in functional responses may also relate to niche separation between the decapods when they occur in sympatry. Our data provides novel evidence on the impact of two established invasive crayfish species in Africa. All the decapods can and will readily consume benthic fish, but however, crayfish impacts will be more pronounced due to the high functional response ratios. The per capita differences between the crayfish and native crabs seen at higher temperatures may contribute to heightened success of the crayfish species, as well as causing biodiversity and biomass declines of native species in the invaded systems. Understanding crayfish impacts on specific ecosystem services is necessary not only for their regulation and management, but also to guard against detriment to human well-being in a continent where food security and water resources are already precarious. Future studies should prioritize the integration of field and laboratory data, especially concerning abundance and distribution. I would like to thank the NRF SAIAB, DSI NRF Research Chair in Inland Fisheries and Freshwater Ecology, VioLab Research Team, DEA, FSDESTEA, MTPA, AFS, and Rhodes University. Thank you, ladies and gentlemen, for listening to my presentation.